Good morning, everyone. Myself, Dr. Sakshi Bharadwaj, on behalf of Elton Technologies, I welcome you all and thankful to have you all here in this one more interesting session of our Women's Day special webinar series. Today's session is entitled as Pipeline Study on Phytochemicals and Nutraceuticals Targeting SARS-CoV-2 and in silico analysis. There is a saying that a sustainable agriculture is one which depletes neither the people nor the land. Natural resources are plenty for us. We haven't utilized them to their full potential actually. With so much evidence of depleting natural resources, toxic waste, climate change, and rapidly increasing instances of natural disasters, why do we keep perpetuating the problem? Why do we continue marching at the same alarming beat actually? We have all experienced the last year pandemic, that is SARS-CoV-2. People have suffered a lot, and I believe that they are still dealing with some consequences. It's a severe acute respiratory syndrome, actually, which caused a global pandemic and is still a global disaster. Naturally occurring compounds are found in plants, and many of them have been studied for their potential antiviral properties and many more other properties. Nutraceuticals, basically the products which are derived from food sources that provide health benefits beyond basic traditional functions. Some phytochemicals and nutraceuticals, such as spices, herbs, fruits, pulses, millets, and other supplements, have the potential for immune boosting and shown antiviral, antioxidant, anti inflammatory effects too. Many traditionally used medicine plants were explored for their bioactive phytochemical used medicine against the coronavirus actually, that is the protein target. Okay, our expert of the day will be going to take us on a tour of how they have used these phytochemicals or natural source compounds from the plants against this particular virus, that is the SARS-CoV-2 virus. Okay, it's giving me immense pleasure to introduce our speaker of the day, Dr. N. Saranya, Assistant Professor Bioinformatics in the Center for Plant Molecular Biology and Biotechnology, Tamil Nadu Agriculture University, Coimbatore. She has completed her post graduation and doctoral studies in bioinformatics at Bharatidasan University, Trichupalli. She has done her postdoctoral studies at Molecular Biophysics Unit, Indian Institute of Science, Bangalore. Dr. Saranya's research interests mainly focus on understanding the intermolecular interaction and the molecular conformations that provide knowledge on biological functional mechanism that a system level through the computer-aided drug designing approach. She has worked on thousands of phytochemicals and nutraceuticals and assisted their efficiency towards SARS-CoV-2 drug targets. This research aided in the food recommendation for the COVID patients during the pandemic in joint with the Government Yoga and Naturopathy Medical College and Hospital, Chennai. Dr. Saranya has expertise in analyzing the genomics, transcriptomics, and metagenomics data as well. Nevertheless, her group has developed a user-friendly, freely accessible Nexa Hub pipeline meant for the analysis of transcriptome data for the benefit of the biological community, which has received the copyright recently. So now, without taking much time, I would like to hand over the platform to our expert. Over to you, ma'am.
Thank you, Dr. Sakshi, for a nice introduction. So, good morning to all of you. So, I'm here to present some of my research on the phytochemicals and nutrachemicals towards COVID targets. So, we many during COVID time, many research groups they were focused on a particular phytochemicals, nutraceuticals, which to come out of that pandemic situation. So in that line, we have analyzed. So from the agri background, so we had the knowledge of many of the phytochemicals and the from the medicinal plants and also the nutraceuticals, which induced us to do this research work, and it got benefited by many of the public people, this research, even though it is just the in silico analysis, not the confirmed by any of the experimental methods. It had its own impact, and we were very happy about the foods that we recommended using our analysis recovered many of the COVID patients. So I'll explain you regarding these works. So I'll discuss these three topics in detail. That is phylogeny of coronaviruses, ritual screening of the 744 phytochemicals from 37 different plant species for specific COVID-19 targets, and effect of these phytochemicals from Indian based functional foods that are used day to day and their impact on SARS-CoV-2 targets like main protease and spike glycoprotein. So I'll be discussing on these three topics today. So COVID-19, so it's one of the unforgettable one, which that is was reported in December 2019, and it had that is the pandemic thing was reported by the World WHO, and it threatened many of the human lives and caused many huge economic losses throughout the world. The only at that time to manage the situation was the lockdown. Lockdown was the only thing, but people don't know how to come out of that situation, and many things were done, and vaccination was found as one of the solution, where many people first priority were given for the aged people first, and then the next government employees, and next only the children alone were left to save the life of many people. The vaccination was the only solution at that time. But even though many of the people were able to survive the infection, even though they were not that is they are not much towards the symptoms of COVID-19. So one of the reason maybe that is those peoples, that is when you compare with the other countries, in India the people were mostly that is less affected. We cannot tell less affected, but we were far more better when compared to the other countries, the death rate and other things. So that time, all through the globe, they turned towards our food, day-to-day food what we take, and other things influenced the other people. So people started investigating what are the food components and other things that supports the immune system to recover from many of the pandemic situations like this. So India also is rich in, very much rich in the plant diversities and also the medicinal compounds. And we are we back around 5,000 years ago towards our traditional medicines like Ayurveda, Unani, Siddha. So those are the basic concepts which led through for the drug development that is happening now. So those ideas and the concepts are the basis for the drug discovery, computer drug discovery, what is now we are doing now. So so what we were interested is we looked towards the phytochemicals, that is the metabolites within those medicinal plants, which will help the recovery of the people. Because the one thing why we went for vaccination itself is no other solution was there, that is no drug was working effectively against this virus, and the mutation ratio was also, it was spreading very high. So the possible drugs that are available were very less, combinations of drugs were used, but then also the recovery rate was very less. At that time, the most of the things most of the people were towards the Ayurvedic, Unani and Siddha medicine. And I think you also remember about the Kabasura Kudineer and other things that were recommended by the arsenic album, those things recommended by the government. So those were was we were able to control or prevent the spread of the disease using those preventive measures. So So first, before the start of the research in the year that is 2020, that is 1920, in that period, we studied the genome of the coronaviruses, because whatever we call SARS-CoV-2, actually it is, that is this virus, the it's phylogeny, that is the previous versions of this virus, which is SARS-CoV, MERS, Middle East respiratory syndrome, MERS, CoV. So like that, many coronaviruses has existed previously. So we want to know how this SARS-CoV-2 is related with that, because other viruses did not cause that much impact when compared to that of the SARS-CoV-2. So we took all the genomes of the coronaviruses that are available in the NCBI database, genome database, and we compared the genomes. So at that time, we were able to see the separate clustering of the SARS-CoV-2 in comparison with the other coronaviruses. But the one thing what we were able to know is this coronavirus, that is SARS-CoV-2, is around 96 points similar with that of the SARS-CoV. It's other variant, and it is around 76.84 similarity with that of the bad coronavirus. So we were interested to compare the that is that is the SARS-CoV-2 with that of the main, that is main protease protein alone, because there are many proteins within available within the SARS-CoV-2. In that, as main protease was a main target studied, so we compared the main protease protein of the three genomes, and what is the difference that is available in the main protease of SARS-CoV-2, so that it is so infective. So we were able to find many regions which are variable among the three SARS-CoV-2, then SARS-CoV and MERS-CoV. So those main protease sequences, main protease protein sequences we have compared, and we were able to find the variable regions, and one variable region which falls in the binding site also we were able to identify, and in that we were able to find that this SARS-CoV-2, it had the alanine in the place of the serine in the case of SARS-CoV, and in the case of MERS-CoV, it was, that is only the alanine was present, but other variable regions has also impact on the infectivity and also the conformational change of the proteins, so that the reason for why this alanine change in SARS-CoV-2 alone is infective, and it's not infective in the case of the other, that is MERS-CoV, is the reason may be the changes that has occurred in the other regions of the protein in the MERS-CoV. So this was the finding we just observed when we compare the genomes and also the sequence of main protease from the other coronaviruses. So with this insight, we started exploring the genome with the many proteins. There are many proteins, that is non-structural proteins were occupying the most of the genome, and the envelope proteins and also the membrane proteins, coat proteins. They were so these are all the proteins they were targeted during that period, so that makes the virus SARS-CoV-2 ineffective. So you could see the most proportion was for the non-structural proteins like NS1, NS2, NS3 up to 12, it was there. So we next step we move towards whether which are all the proteins that has the experimental structure. So at that time we were able to find some 7 targets from SARS-CoV-2, And for main protease alone, we had the structure of the MERS-CoV and SARS coronavirus. So all these proteins were targeted. So we choose all these proteins, and their PDB IDs are given here. We chosen all these protein targets in order to understand their mechanism of action towards the phytochemicals we have chosen for this study. So, and also we have compared, so the proteins are the folded structures in its structural and the functional form. When it gets folded, so we have seen the sequence similarity of the MERS-CoV and SARS-CoV-2 main protease. You can see the most of the regions are similar, but the variation in the certain regions that makes the functional difference. If that is going to be the case, see here the binding site, the conformation or the binding site volume which accommodates the phytochemical or any of the ligands, small molecules or the inhibitors, they also vary largely for the same protein, like in a different coronaviruses, and also for the other proteins of the SARS-CoV-2. It varies largely. Their binding site is completely has different conformations. These all minute, the different conformational minute change in even a single residue affects the binding of any drugs, small molecules, ligands, or the phytochemicals or nutrachemicals you are studying. So in that case, it's very important to understand how does these proteins interact with the phytochemicals or the nutraceuticals we are choosing for the study. So in that way, we have done the docking and the virtual screening experiments in order to understand how the phytochemicals from different medicinal plants will inhibit the activity of the chosen targets. So, my second topic, the virtual screening of 744 phytochemicals from 37 different plant species for specific COVID-19 targets. So, the targets are, as I have said, it's SARS-CoV main protease, MERS-CoV main protease, SARS-CoV-2 main protease. So, these three are the same protein from different coronaviruses. And from SARS-CoV-2, it includes other proteins also, like spike glycoprotein, RNA dependent RNA polymerase, NSP16, NSP10, NSP3, NSP15, and NSP9. So, with all these structures, experimental structures were available, and we have chosen these structures as a target for the further virtual screening experiment. So, these are the 37 medicinal plants we have chosen based on the previous knowledge, that is, we have obtained through the literature studies which are having the antiviral property compounds which are used in Ayurveda studies, and also in Kabasura Kudinir. So, what are all the ingredients or the available, which plants were extracts were used, and so from the Siddha, what are all the medicines that are reported for antiviral. So we compiled the most prominent ones, those 37 medicinal plants we have chosen, and these phytochemicals, the phytochemicals from these 37 medicinal plants we collected from the Dr. Duke's database and PubChem database, which were used for our experiment. The 37 plants which we have chosen is the Ableton indicum, Acorus calamus. So in the bracket, I have given the Tamil name of all the medicinal plants, and Egli marmilis, then Anacamis pythium, Andraglophis paniculata, Asaradicta indica, Brassica oleracea, Carica papaya, Cissus quadrangulis, Clerodendron serratum, Cocos nucifera, Coriandrum sativum, Cucurma longa, then Cypress rhodentis, then licorice, which is a Glycyrrhiza glabra, then Hydrophila auriculata, then Ipumia carnea. So like this, 37 Piper beetle, Piper longum, then Plectranthus ambionicus. Like this, we have Terminalia billerica, Terminalia turbula, Like this, we have chosen 37 plant species from which the phytochemicals were collected, and then we have further carried out the docking experiment. So the methodology goes in this way. First, we have done the literature review, and the biological databases were used for the collection of the phytochemicals from the databases like PubChem and Dr. Duke's database. So, as I said earlier, the protein targets, that is the proteins which we have, SARS main protease, spike protein, then non-structural proteins. Those structures were collected from the RCSB PDB database, and though for those proteins, the binding sites or the active site of the protein targets were identified using the CAST-P server, and the proteins and the phytochemicals which we have identified, or we are going to find out their antiviral property, were docked against the protein, and we have further interaction analysis was performed based on their docking score and also which residues it is interacting in the binding site. So those analyses were carefully done in order to understand what all the phytochemicals that really can have some impact on the function of the protein targets. So the first thing, the target, what we main protease, which we are interested, we took, and we have done the virtual screening experiment of all the 744 phytochemicals, and we were able to find that a compound from gingiber officinale, it's ginger actually, the cyanine from this particular gingiber officinale was found to have higher docking score or binding affinity with the main proteases of all three coronaviruses, and which is placed in top 10 hits in the list when we screen among all the 744 phytochemicals. So this was the interesting observation we could make from the results. And the binding affinity value was found to be minus 8.3 kilocal per mole, minus 8.2 kilocal per mole, and minus 7.7 kilocal per mole against SARS-CoV-2, SARS-CoV, and MERS-CoV main proteases, and their hydrogen bond interactions, and which residue it makes the interactions. So those details you can observe from the image. And also you can see, even though those three proteins are the same protein but in different viruses, the binding mode of the cyanine phytochemical, it was totally different, and it was binding mode was the conformation was different, and the after binding or before binding also, the binding site size or its volume was very largely, and you can see how the residue changes are while the interaction is happening. So the next finding was that there was certain compounds like amantoflavone, augustus flavone, which was inhibiting, that is most of the targets of the SARS-CoV-2 like NSP12, NSP9, NSP15, NSP10 to NSP16, and spike glycoprotein. So this compound, which also had many other biological activities like anti-tumor, antiviral, anti-inflammatory, antioxidative, anti-diabetic, and antifungal, so on. I have limited to certain biological activities alone. Otherwise, the list is going to extend. In the same way, the Augustus flavone, which is also a flavonoid, so it also had the binding affinity value of minus 8.9, minus 8.2, and minus 6.6 towards different targets, NSP, that is non-structural protein, and SARS-CoV-2 main protease. So this was the main thing with the amantoflavone and augustus flavone. They had a very good inhibitory activity based on the docking studies results we were able to observe, and we reported in our publication. So, and the next thing is, the scientists were also interested to know what are the drugs that are prevailing already approved by FDA, which can inhibit the SARS-CoV-2 targets. So, what at that time, the mostly, that is during the COVID pandemic, we were able to find the three drugs which were mostly used for the this treating the COVID patients, that is hydroxychloroquine, chloroquine, and ivermectin. So we have performed the docking experiment on these drugs also to understand whether how far their binding affinity is for the targets that we have chosen compared to the phytochemicals what we have got as a result. And you can see from the results, as observed with the COVID patients, the ivermectin, which was used at that time, compared to the hydrochloroquine and chloroquine, the ivermectin showed the higher binding affinity results against all the targets compared to that of the hydroxychloroquine and the chloroquine compound. So the results were most favorable to whatever happened, that whatever the drug that was recommended during that period. So this results and their binding mode, their mode of action, how do they interact, their binding affinity value, this all interpretation, we came to the conclusion that ivermectin it performed better on all the COVID-2 targets when compared to the hydrochloroquine and chloroquine. And we were also interested to know how the plants, that is which plant is having most effective against the SARS-CoV-2 targets, that is which plant compounds are able to inhibit most of the SARS-CoV-2 protein targets. So this analysis we have performed using constructing a network diagram. So you can see here the green circles represents the protein targets, and those are connected with the plants, the phytochemicals from the plants which inhibits that particular target. Okay, so in that case, the NSP15 was most inhibited by many of the phytochemicals from different medicinal plants, and NSP9 non-structural protein, then RDRP, NSP10 to, and so these in the order. So maximum the inhibited one is NSP15, and the minor inhibited the phytochemicals from the medicinal plant species are SARS-CoV-2 Mpro. Likewise, the plants which maximum had the phytochemicals that will inhibit the targets are Vitex nugindo, then Solanum niger next place, Terminala tubula, which is the turmeric one. So it is in the third place. Likewise, the least one is the Nigella sativum, then Azadirachta indica, like that, Malus domestica. So we were able to categorize which medicinal plants are having more effective against in the inhibition of the protein targets, and which plants are contributing more antiviral compounds that could inhibit the SARS-CoV-2 targets. The pink ones are the other FDA approved drugs and the antiviral compounds which were also taken for this particular experiments and which has shown the activity. Of course, the antiviral compounds it stood in the fifth place when compared to the phytochemicals from the medicinal plants in inhibiting the SARS-CoV-2 targets, and the FDA approved drug also it stood behind when compared to the other things, that is Magnifera indica. So many medicinal plants they had many phytochemicals which were able to inhibit the activity of SARS-CoV-2 targets. So this study was only based on the phytochemicals from the medicinal plants. Then we were interested to know what role the phytochem, that is as Indian cuisine or the Indian foods have gained much importance in the global scenario. We want to know how our food habits or how the food, what are all the that is metabolites within the food that make the body immune system better, or to just prevent against many of the viral diseases, so that the immune system can withstand from any of the diseases. So we we were able to find at that time itself many research papers on the one of the food what we used to use in that is the rasam. in rasam, what are all the ingredients added, and how much it is. You don't believe it was the rasam was tested for its anti-cancer, anti-tumor, antibacterial, anti. So many research papers have come. If you just search in the Google Scholar or the PubMed literature, you can get much more information regarding how the Indian foods are used. What is the importance of that in many of the disease conditions. So in that ways, what we have done is we have so many groups are focusing on limited foods or limited that is plants like that. So we as a whole, as how much information that we could collect, we have collected from the food based on their cat. We have divided it into different categories, and we have performed the how those food metabolites have their role in the inhibition of SARS-CoV-2 targets, main targets, that is the main protease and spike glycoprotein, core protein. So those things, those we have focused on only those two protein targets for this analysis. So we have taken the foods like vegetables, which included 15 plants, fruits for 11 plants, and supplement it's 3, and spices 7, pulses 4, millets 3, and herbs 20. The list of the plants are given here. So fruits, it includes apple, banana, gooseberry. That's whatever we use in common, and for how far we can get the information of those things. So everything was included, and totally around 63 food crops with 4462 phytochemicals was taken for analysis. In millets, the metabolites which were that is proso millet, pearl millet, and finger millet. So those were used for the phytochemical obtaining the phytochemicals. And the pulses, which is includes the groundnut, rooted green gram, black gram, PGNP. So those pulses are used. And the herbs, which includes fenugreek, nunalis, piper beetle, vallare, wheatgrass, adimaduram, acarus calamus. So I'm randomly speaking about the names. The spices, which includes curcuma longa, cinnamon, piper longum, cardamom. So whatever that we were able to collect in vegetables, mint leaves, solanum tarum, snake gourd, coriander, bitter gourd. So whatever the phytochemicals of the regularly used food, functional foods that we use, so that we have collected, and we just screened the antiviral compounds. So for this experiment, what how we have performed is the same protocol of collecting the phytochemicals from the databases and also from the literature. And the protein target structure, only two protein targets we have chosen: spike glycoprotein and main protease. And we have just done the docking, and also we have done the analysis. So here we have not taken that is we have for which phytochemicals are considered as antiviral, means those phytochemicals upon docking which is having a binding affinity value of -5 and lesser than that, because whenever the docking score is -5 or in negative, it means that it binds stronger. So we have set a threshold that the docking compounds, whichever is going to have the -5 and less, are taken for the further analysis. So here I have projected a table which will contain the phytochemicals from different food categories, and it look first three compounds which are having the higher docking score, and the interactions are alone taken for the that is reported in this table. So you can see here some of the compounds, they are present in which plant, and what is their binding affinity, what is their pubchem id, and from which part of the plant those phytochemicals are available. So those informations are displayed in this table, And you could also find some of the phytochemicals. It's not that one phytochemical, if it is present in one plant, it should not be in other plant, right? So in that case, the vegetables, so that olenolic acid which is present in cyperrodentes, it is also in the eucalyptus, osmum bacillum, osmum teniflorium florum. So like that, so the compounds may be the same, but the source from which they come is different. And based on their category, we have listed the first three compounds from the first three plants. So Cypress rodentis, Allium sativum, and then Moringa oleifera. So these were the first, the phytochemicals were reported from this plant. For the spices, the catecholic acid, gingiferol, then gamma eudestomol, and secosterpenes. So these are the compounds that is reported for spices. So here, if there are compounds which have the same docking score and the interaction, similar kind of interactions, so those compound also the first three places which compounds occupies if same binding affinity is retained by more than one compound, we have not missed that compound, we have included that also. That's why the four compounds are reported in the spaces. And in the case of millet, it is violanthin, naringenin, and orientin from finger millet were reported. So these result is the docking results of for the target SARS-CoV spike protein. For the main protease, I will show you the table in the next slide. And for the herbs, you can see here. And for the pulses, it was from Arcus hypogaea and Kanjanas kanjan. So although we have taken many pulses, you can see the antiviral compound, which is of high efficient, was reported from the same plant which is present in the seed. And then Kanjana's Kanjan, which I have Kanjanun. So the supplements, the ghee, caronin was reported, and you can see the from the fruits, from Mitis vinifera grapes, it had oleanolic acid, then citrusin from Citrus sinensis, and Phyllanthus emblica. So it had a corallagin compound. So this was minus 12 was the largest, that is the highest binding affinity or the docking score we could obtain for the mucinic acid, the corallagin. So these are the compounds which are able to inhibit the spike, the spike protein with highest efficiency. So next, the remaining compound citrusin, that is citrus sinensis, which is orange. So like this, the compounds are reported for different category of foods. So next we will move to the slide, and here you can find the top three phytochemicals which are having the lowest docking score for the SARS-CoV-2 main protease. So here you can see the same compound sometimes is repeated, but they were able to inhibit the protein, that is with a stable interaction. So here we have mainly looked for the interactions like hydrogen bond interaction, hydrophobic interaction, and van der Waals interaction. We can see the binding affinity value of minus 12.7, which is the highest binding affinity reported for the oleanolic acid for the target, the main protease. Likewise, this catecholic acid, which had minus 12.3, then 13 was minus 13 was reported from Azadirachta indica, which is a nimbine compound. Then 12.9 minus 12.9 binding affinity was observed from the leaf of Azadirachta indica, that is nimbindiol. So like this, so you can see the minus 15 for retroid for citrusin, and I hope you can recollect that citrusin was reported for the spike glycoprotein also. So Phyllanthus emblica, that's corallagin. So likewise, we were able to vitexin from finger millet. So you can see, although we have taken three millets for this study, only finger millet was able to report the highest binding affinity compounds, which can inhibit the SARS-CoV-2 targets. So in this way, we were able to identify the phytochemicals or metabolites which are able to inhibit the SARS-CoV-2 targets. So here is a bar chart which will give you the overall picture of how many phytochemicals are present in each food category, And how many are known to inhibit the main protease? That is, the orange one shows the phytochemicals in a particular plant which were able to inhibit the main protease, and the gray one shows how many phytochemicals can inhibit the spike glycoprotein. And you can see the fruits, they have the maximum antiviral compounds. The compounds that are present is also maximum, and their antiviral nature is also maximum when compared to the other food categories. But in vice versa, if you compare total number of compounds and the total number of compounds that are inhibiting, you can see there are, even though there are many compounds, only some of the compounds are going to have the antiviral nature. But in the case of millet, the compounds are less, but those lesser number of compounds, they have the antiviral property. So we were able to find that aspect from this graph. And so you can see here the fruits, vegetables, and herbs, spices, they have the maximum number of antiviral compounds when compared to that of the supplement, millet, and pulses. So these results were given to the government yoga and naturopathy medical hospital in Chennai, where they recommended the foods based on the antiviral nature, and they created a menu accordingly. And they reported us that, yeah, based on this results, they framed this food chart, and they were able to see the recovery of patients based on the food things, food menu they have followed. So for this work we have, so it's not completely a teamwork which we have done along with the students and colleagues to perform this analysis. So you can see here the antiviral compounds that are the number, based on the number values here, the information is given from the grapes. Some 193 phytochemicals, they had the antiviral nature, and it inhibited the main protease. And some 174 phytochemicals, they inhibited the spike glycoprotein protein targets. Likewise for different things. So, based on the results, fruits, vegetables, spices, and herbs are the categories that possess a higher number of anti-COVID phytochemicals than compared to the pulses, supplement, millet. So, this conclusion we were able to derive based on the analysis what we have performed. And here, for this, that is food phytochemicals also, we have performed a network analysis to understand what are that is phytochemical wise which compounds are having the ability to inhibit both the targets, that is main protease and spicoglycoprotein, and which are the compounds that have the efficiency to inhibit only one target. So these are all the riboflavin, caronin, sequiterpenes, gibberellin, then collagen, then catecholic acid, olenolic acid, diterpenoid, epoxygen, and citrusin 2. So some of these compounds, they showed the possible inhibition of both the targets, whereas the other phytochemicals, they were able, naringenin, mesylenic acid, orientin, gingiberol, flavonoids, tocco. So other compounds, they were able to inhibit only one particular protein target. So, with this thing, I would like to end my talk. And this is my group. So, this group, it contains that is there are my colleagues and also my students, and he's my son. So when we went out, we just took this picture. So I'll be so this is a reference. So this is a book chapter which is published recently, and this is the article which I had covered the most of the portions for this lecture, which was published in Frontiers in Pharmacology. And now I am happy to take the questions.
Yeah, thank you so much, ma'am. It was very knowledgeable session actually. So we have seen how you have selected the different phytochemicals from the different sources, and how you have concluded with your results. Of course, even we have seen in your results that the how fruits basically from the vitamin C component those are having are helpful of secured or inhibit that SARS-CoV-2 virus basically. So yes, now it's time to take some questions. Okay, the first question is how to identify the immunoboost phytochemicals.
Okay. So, the phytochemicals that have higher antiviral compounds, we can have an idea of those foods as the immunobooster phyto there is foods. That means, so based on our analysis, we are able to find which foods are having higher phytochemicals. So based on that only, they have recommended for that is the government of Ayush, that medical hospital also they recommended the food. So those food that have many antiviral compounds, of course, it may have the immune boosting ability also. So yes, the food that we take actually is having is enriched with antiviral compounds. So we can tell all the foods that we have are having the immunoboosting properties.
Correct. Adding to that, ma'am, as you have given the example of rasam, each Indian food is full of many different, I would say, the properties. What is basically that's why our elders are, already have invented those kind of dishes which we are being used nowadays. So yes, definitely. Next question, please. How to construct the network of compound with plant?
So we have used the yes, ma'am.
How to construct the network of compound with plant?
Yeah. So we have used certain softwares like Cytoscape in order to construct the network, which we were able to identify which are all the compounds or having the common activity. Because just by having the thousands and thousands of phytochemicals and with the values, we could not interpret the results that much easily. It was not that much easy, even though the experiment it went in silico, the analysis and interpretation it took months, and because we have to support our findings and we should also justify the compounds that we report and the plants that are reported. So everything, each and every step we have carried out carefully, and the network of compound, it was the last part which we have done. So those compounds which were able to inhibit both the compounds, both the targets, we have taken and we have constructed a network using the Cytoscape software.
So I request you, ma'am, if you could able to tell some, servers from where my viewers can download the, those data banks of the natural compounds. That would be great. Means helpful for them. So if there
Yeah,
any.
so the databases what we have used is freely accessible databases. Dr. Duke's database. So in Google, if you are able to search Dr. Duke's database, so it will take you to that page, and whatever plant phytochemicals you need, you can give the search. Just like a Google search, you can give the plant name. It will list the number of compounds within this that particular plant. So how we so there were many plant paths that were available, and most of the plant paths which we are interested and which were used as a medicine in the earlier period, so those paths alone we have chosen, and those phytochemicals we have downloaded and we have chosen for this analysis. Dr.
Okay.
Duke's and PubChem database. Sometimes the structures are not available. Only the name of the compound is available in Dr. Duke's database. So the structure of the compound we can obtain it by using the PubChem database. If you just give the search of that compound in the PubChem database, you can get the structure from the PubChem database. NCBA PubChem.
Okay. Thank you so much, ma'am, for answering that question. Do we have any other question? Okay, I think we have done
Thank
with
you,
our
Dia.
questions. So thank you so much, ma'am. It was really knowledgeable with the natural, how we can take those natural sources to being away from all those kind of disease condition, because each and every food content, whatever from are the natural sources, are being helpful for us to, take the advantage for our health, of course. So it's time to now felicitate our speaker of the day with a memento and a certification of appreciation. So thank you so much, ma'am, being with
Thank you
us
so
today
much.
for this
So,
presentation. It was
yes,
a
ma'am.
pleasure to have you there with us. Yes,
And I would like to end my talk with that the food, they are the more they are more like medicine when compared to the medicines that we take. So
correct.
when we have the proper the food without neglecting many of the things, that's enough for us to stay
Yes,
healthy. It
yes,
prevents from many of the diseases when compared to just going behind any of the medicines for any sake. It's the food are the medicines, and it has to be taken care. So as a woman, we have in our hand our family health and everything. So that I would like to conclude.
that's true, ma'am. That's true. Appreciated, yes. So, guys, stay tuned with us. Please subscribe us, like us, and follow us for upcoming events. Let's connect and grow together. Thank you. Thank you all. Thank you so much.
Thank you, ma'am.
Thank you.